Hello and welcome to second episode on Archaebacteria. This is a part of the series called Know Your Kingdom. Have you ever heard of this group of microorganisms? Well, they are unique and fascinating creatures that have captured the attention of scientists and researchers for many years. In this video, we will explore the world of Archaebacteria and discover what makes them so special. Next episode will be on Eukaryotes. Make sure to subscribe for the same. So, let us begin with what Archaebacteria are. Archaebacteria are a group of microorganisms that belong to the domain Archaea. They were first discovered in 1977 by Carl Woese and George Fox and have since been studied for their unique cellular and metabolic properties. Archaebacteria are found in a variety of extreme environments, including hot springs, hydrothermal vents, and saline ponds, where they are able to thrive in conditions that would be lethal to other forms of life. Unique characteristics of Archaebacteria. Archaebacteria have several unique characteristics that set them apart from other forms of life. For example, they have a different type of cell wall structure, a different type of RNA, and different types of membrane lipids. Additionally, they have a different type of metabolic process, called methanogenesis, which involves the production of methane from carbon dioxide. Are you listening to as a podcast or viewing on YouTube as video? Do comment. Surviving in extreme environments. One of the key features of Archaebacteria is their ability to survive in extreme environments. This ability is largely due to the presence of highly specialized structures and processes within the cells of these microorganisms. For example, many species of Archaebacteria have a unique cell wall that is composed of a material called pseudomurine. This material is more resistant to high temperatures and high levels of salt than the cell walls of other forms of life, which allows Archaebacteria to thrive in extreme environments. Let us discuss it in some detail. Pseudomurine, also known as pseudopeptidoglycan, is a type of cell wall that is found in certain groups of Archaea. It is similar to the peptidoglycan cell wall found in most bacteria, but contains different sugar and amino acid building blocks. Pseudomurine is composed of N-acetylglucosamine and N-acetyltalosamine acid, which are connected by beta-1,3 linkages. Do not worry if it sounds complicated. Nature is the most complex simulation after all. These linkages in particular are similar to those found in fungal cell walls, but are different from the glycosidic linkages in peptidoglycan. The unique structure of pseudomurine helps Archaea to survive in extreme environments, such as high temperatures, high salt concentrations, and low pH levels. Moving on. Methanogenesis. Another feature that sets Archaebacteria apart from other forms of life is their metabolic processes. Many species of Archaebacteria use a process called methanogenesis to produce energy. In this process, they use carbon dioxide and hydrogen to produce methane, which they can then use as a source of energy. This type of metabolic process is not seen in other forms of life, which makes it a key characteristic of Archaebacteria. Let us talk about the role of Archaebacteria in the global carbon cycle. First, let us understand what this cycle is. The global carbon cycle is the process by which carbon is exchanged between the Earth's atmosphere, land, oceans, and living organisms. Carbon is continually cycled through these different reservoirs in the form of carbon dioxide, a greenhouse gas that plays a critical role in regulating the Earth's climate. Human activities, such as burning fossil fuels and deforestation, have significantly altered the global carbon cycle, leading to increased levels of carbon dioxide in the atmosphere and contributing to climate change. Understanding the global carbon cycle is essential for developing strategies to mitigate the effects of climate change and ensure a sustainable future for our planet. Archaebacteria are also important to the global carbon cycle because they play a key role in the production of methane. Methane is a potent greenhouse gas, and the production of methane by Archaebacteria contributes to global warming. Archaebacteria are found in large numbers in environments such as rice paddies, landfills, and the digestive tracts of ruminant animals, where they play a key role in the production of methane. What is the future of Archaebacteria? These have been the subject of much research in recent years due to their unique properties and abilities. Scientists are interested in learning more about these microorganisms and how they are able to survive in extreme environments. Additionally, researchers are exploring the potential of Archaebacteria for biotechnology applications, such as the production of biofuels and the removal of pollutants from contaminated environments. They have unique biochemical pathways that allow them to produce methane, hydrogen, and other chemicals that could be used as biofuels. Archaebacteria are of interest to researchers looking for sustainable sources of energy, as they are able to produce biofuels using carbon dioxide and water, which could potentially reduce greenhouse gas emissions. However, there are still challenges to be overcome, such as finding efficient and cost-effective methods to cultivate and harvest these microorganisms for biofuel production. Next episode will be on Eubacteria. Make sure to subscribe for the same. These unique organisms also have shown potential in removing pollutants from contaminated environments, such as wastewater and soil. Archaebacteria can break down pollutants such as hydrocarbons and heavy metals through a process called bioremediation, where they use enzymes to convert the pollutants into less harmful substances. This natural process is both cost-effective and environmentally friendly, making it an attractive option for cleaning up polluted sites. In conclusion, Archaebacteria are a unique and important group of microorganisms that play a critical role in the global carbon cycle and have unique features and abilities that set them apart from other forms of life. These microorganisms are the subject of ongoing research, and their study will likely continue to provide important insights into the workings of the biosphere and the potential for biotechnology applications. Next episode will be on Eubacteria. Make sure to subscribe for the same. Thank you for listening to this podcast video on Archaebacteria.